Invitation. So it's a great pleasure to present today the work and more broadly on technologies in medicine's quality on behalf of the Medicine Quality Research Group. We are working on very different aspects of the problem of poor quality medicines. But that one is one I started to work on very early when I joined the group, then based in Laos in Southeast Asia. So I will just first give a little bit of context first for those who are not too familiar with what are the problems of poor quality medicines. Just a little bit of history. So this is a problem that has a long history, actually. We have heard about quality issues, in particular falsification, since a very, very long time. So in ancient Egypt, there was expeditions from the Hatshepsut, the Egyptian pharaoh, to obtain genuine herbs because he had suspicious on the herbs she was using. And then in AD 50 to 70, the Greek pharmacologist, chemist, and botanist Dioscorides wrote a book called De Materia Medica. And this book actually explained, it was a precursor of modern pharmacopoeias because it explains how to use different senses, taste, odor, and colors and feels to identify whether substances were good or not good quality or falsified. And as soon as the trade of herbal products started, especially the one of cinchona bark, which is a plant that was used to extract quinine, the first ever antimalarial, in the 17th century, there was a big fake trade of this, and the barks were adulterated with other species of barks. And then when the first remedies in the 19th century started to appear, herbal remedies, there was extracted from herbs. Fake quinine trade also appeared on a quite large scale, and quinine vials were actually containing salicylene, which is a derivative from salicylic acids, or sometimes it contained face powder and other different things. So there are some articles on this. And sorry, I cannot see well on the screen. Okay. And after in post-war era, there was also a big markets of fake penicillins. 7 people were arrested in Berlin because they were trading with fake penicillin that contains vials contain glucose or crushed antimalarial tablets. And there were also a movie around post-war Vienna issues of also fake penicillin called The Third Man that is very interesting movie if you haven't seen it yet. But that's about the fake trade of penicillin in post-war Vienna, where there were issues at that time because penicillin was already produced and distributed, but was controlled by American and British army and used mainly for the army officers, but other people needed it. So there was a black market, sometimes borrowing the sewing system to distribute the medicines, the fake penicillin. So that's quite interesting movie if you're interested in the topic. Okay, let's go to a more recent history now. Sorry, I cannot change the slide easily. Okay. But first let's have a look at what we are talking about. So we like to use this Venn diagram in our group to explain what we're talking about because the quality of medicines is quite a broad problem. Different quality defects can be seen, and we tend to classify poor quality medicines into 3 different categories. So if you look at the black circle, that is all medicines that are not good quality. Then we have substandard medicines that are manufactured by authorized manufacturers, but they poor quality results from negligent factory errors. So they are different from falsified medicines that are made by criminals. So there is an intention in it to copy a medicine or to make a falsified version of a medicine. And we also have medicines that are degraded. So they are good quality when they leave the factory, but they can degrade during transportation or during storage because of the heat, humidity, and other factors. And so falsified medicines, I forgot to mention, can contain no active ingredients, or they can contain wrong wrong active ingredients. Sometimes can be toxic, or they can contain a little bit amount of active ingredients. There is a big diversity of what falsified medicine can contain based on the criminals' work. Substandard medicines, there's also a lot of different defects, quality defects that can be found. There can be issues with the quantity of active ingredients. There can be dissolution problems because the excipients were not the right ones, or the manufacturer there was a problem during the manufacture. And degraded medicines obviously usually have our lower active ingredient amounts, and they can sometimes have um, related substances also appearing. So that's the diversity of the problem with poor quality medicines. The relative sizes of the circles are unknown, even though they are equal on this Venn diagram. That's not right. We don't really know the extent of the problem, and I will explain this in the next few slides. And also they overlap on this diagram because it's sometimes very hard to distinguish between one or the other. I just wanted to give you a few examples of problems we've seen in the past. Like, that problem, that on the slide, what you can see is one package is a genuine medicine and one package is a falsified medicine. And it's an anti-malarial artesunate that was used as a monotherapy in the past to treat malaria. And this problem occurred in the late 90s when there was a big epidemic of falsified artesunate tablets in Southeast Asia. I was not yet working on this then, but Paul Newton, the head of our group, investigated the issue and found that in more than 80% of the pharmacies in I think 5 different countries, they could find fake falsified artesunate tablets. And these ones contain no active ingredient. They were mainly flour. So, but you can see that the difference between the two is very hard to see. There is actually, if you look at them on a picture, it's not easy, but if you look at them physically, you can see one major change, and this is this little comma on the package of the left that is not present on the package of the right. And if I remember correctly, the left one is the genuine medicine and the right one is the falsified version. Just to mention that the criminals here had even falsified the hologram that you could see in quite a good way for some of them. There were different versions of these holograms through time at that time, interestingly, different fake holograms. But they were quite good. And if you didn't know how to look whether they are fake or not, that was really not easy. A few more recent, though actually quite old now, example of this time. I think we could call it a substandard medicine, but that's sometimes, as I mentioned, it's a good illustration of when it's difficult to make the distinction, and I will explain why. So that's the Lahore disaster in Pakistan, when 200 patients from the cardiology department of a specific hospital in Lahore died because of bone marrow failure. And the common points between these patients were that they were taking isosorbate mononitrate 20 milligrams, which is a medicine used for a specific cardiovascular condition. And what happened, the story in short is that this isosorbide medicines contain excessive dose of pyrimethamine, which is an antimalarial that is very well known for its bone marrow side effects, toxicity on bone marrow, sorry. Um, and what happened in that case is that it's multifold. There was a lot of factory errors. The first one, the first striking one, was that at that time there was a flood in the port where the barrels of the isosorbide raw ingredients were stored. So there was flooding, and so the barrels were affected. So there were the powder inside the barrels was moved into new barrels, but where the new barrels were not relabeled. So there was a mix-up between the barrel of isosorbide and pyrimethamine raw ingredients. But then this error was not captured during the whole manufacturing process. There was a lot of different errors. So you can wonder whether it's a substandard actually medicine or was it criminal? There was something kind of criminal negligence in it. So where does that stand basically? There is a very interesting report. It's very rare to have these cases in such detail. So the investigation around what happened, and the report was released by the Pakistani government, and it's called the Pathology of Negligence. So if you want to have a look, it's easy to find online, but if not, just let me know. I can send to you. But that was an interesting case, and there are many other cases, and this one is interesting because we often think about poor quality medicines that contain lower active ingredients that they should contain, but actually that's not all the only problems. In that case, I was in Pakistan again, and the lines represent the incidence of malaria cases per 1,000 persons per year throughout the year, and the different colors represent different refugees camps where there was a very strong monitoring of malaria cases. And you can see that in 2003, the number of cases was really much higher than in the previous year. And so that's when epidemiologists started to investigate what was going on. And the story short is that there was a change of a brand of antimalarial at that time. And the new brands contain actually more than the active ingredients that was stated on the label. And the active ingredients were sulfadoxine, pyrimethamine. So there was almost 130% of what there should be. So there was more. But the dissolution of the tablets was not happening at all. So when you take a tablet, the medicine has to dissolve to reach the bloodstream. In that case, it did not happen at all. So that was going straight to the stools, if I may. So no effect at all. So that's another example. That case was not a falsified medicine. That was a substandard. That was a very bad manufacturing error. So yeah, so I like to give a few cases before starting to talk about the rest, and also people will ask me, but how big? What's the extent of the problem? It's a very tricky question because there is a lot of gaps of evidence. It's a relatively new research field, let's say, and kind of neglected. So there is great need for more and better quality data for interpretation. But you will read, it's a bit old now, but you will often read that 10% of medicines in low and middle income countries are substandard or falsified medicines. And it comes from this report from the WHO published in 2017, where they did a systematic review of all the studies where researchers or regulators had collected samples to test their quality in the last 10 years, so in the previous 10 years, sorry. And that was almost 50,000 samples in total over 10 years, 55,000 samples in total. And they didn't look specifically for low and middle income countries, they include everything. But in the end, they found that most samples were collected in low and middle income countries. And they got these numbers as an estimate of the prevalence of poor quality medicines. But a few things about this number we need to be cautioned about is that First, most of the samples collected in these studies were anti-infectives, as you can see on the table at the top. And also, there's not a lot of samples for 10 years. And most of the studies were done in a convenience approach. So they were not randomizing the selection of the pharmacies where they would collect the medicine. So 77% of the studies included in that with convenience sampling. So the results might be a bit biased. Okay, so let's stop about talking about the context, and let's go into the topic of today. So technologies to detect substandard and falsified medicines. That's what I'm going to talk about today. This is of particular interest to myself and the team, and we work with brilliant chemists and biochemists and physicists as well on this topic. So the WHO strategy to fight against substandard and falsified medicine. Sorry, you will see SF often is the acronym that everybody uses in that field. So that's for substandard and falsified medicines. So the WHO strategy is around 3 different pillars. And one, so there is a prevent and response, and there is also the detect pillar. So detection is quite an important pillar within that strategy, but just please keep in mind that's not the only way to fight against the problem, especially in low and middle income countries. And so in an ideal world, and that's what happened in many high-income countries, there is sufficiently well-equipped laboratories and resources and human resources, including enough trained, and there is plenty of money and time to conduct post-market surveillance of the medicines and the medical products in the supply chains. But that's the ideal world, and unfortunately, especially in low and middle income countries, that's not the case. There is not enough equipment and personnel and not enough money. It costs a lot of money to test medicines to conduct this post-market surveillance. And that's why screening technologies have gained quite interest in, especially in LMICs, to assess the quality of medicines in a rapid, easy, and reasonably cheap manner, to identify suspicious samples. But then it's not meant, as the name indicates, they are not meant to replace compendial testing in the lab. And I like to use this illustration on the right. There are still cases of falsified medicines where you can visually see that there is a problem with the medicine. Sometimes there are spelling mistakes, but it's not always the case, especially with now access to a lot of different technology. Criminals have become quite good at making nice copies of packaging, for example. So detection technologies have gained a momentum. And so what are we talking about? So we are talking about the interest at the moment is done on portable screening technologies. So when you do a search in Google, you will quickly find like dozens or even hundreds of devices that could work for testing a medicine. But many of them, and especially spectrophotometers, were not developed for this purpose initially. They were not manufactured for that. And I will come back to this. There are now new technologies that are being developed for especially for testing medicines, but that's not the majority of them. On the screen is just a subset of what's available. There are really hundreds of them. And there are many shapes, sizes, costs. There are nice looking, fancy gun-sized, gun-shaped, I would say, devices and more smaller, like lower cost and single-use devices. And I will show to you a few in details. Some low and middle income countries already use portable devices for screening medicines, and one is the mini lab. It's been used for decades now. So it's a mini lab, it's portable, but it's two big suitcases of 50 kilograms. And it contains, you can kind of see on the picture, it contains laboratory equipment to conduct a few different things. So there are guidelines on how to assess the visual appearance of medicines. And there is something called TLC, the thin layer chromatography, that helps you to identify whether the active ingredients is present in the medicine, and it can tell you a semi-quantitative aspect as well. So it can tell you whether the active ingredient is present below or above 80% of the amount stated on the label. So that's the main test that is usually it can also tell you, it can also assess whether the medicine disintegrates well in a liquid. It's relatively cheap. And so at the moment it can test 125 different active pharmaceutical ingredients, mainly, if not only, essential medicines, mainly anti-infectives, but there is also anti-diabetics and others. Spectrophotometers are the other technology that is attracting interest. It's been attracting interest in the last decade or two. I'll give you a little bit of like technical how it works a little bit, because it's useful for the rest of this talk. So these are two examples of spectrophotometers. How they work is that the very simplified way to explain is that you shine a light to the sample, as you can see on the top picture or even on the bottom pictures. There is a light that goes through the sample, and then it makes a molecule vibrates. And the result is that the signal that comes back from these vibrations is captured by the device and gives a spectrum that you see at the bottom in red. And this spectrum is often called a chemical fingerprint, and it's unique to that specific vial or tablet. And I go into a little bit more details. And what happened then in the device is that the device will kind of overlap the spectrum from the medicine you test to a reference spectrum here in blue. And then it does the overlap, and then it will say, okay, this fails. It's not the same spectrum. It's not what you expected. It's a fail. That's a very simplified way. Sorry for the chemists or physicists in the room, but that's the easiest way to explain. The problem is with these devices is you need to get the reference spectrum. And there is not a lot of evidence on how you create them, but ideally you need several batches of the same medicine, and ideally produced in different manufacturing plants to have a good, sorry, to have a good, sorry, I lost track, to have a good reference library. And to take into account inter-batch viability. So to illustrate here that what was being done is that so you then merge the different spectrum, and you get the reference library, the reference spectrum. That the TrueScan is one that has been used already since maybe a few decades in some countries. Nigeria, for example, is using them. It's a Raman spectrophotometer. I'll spare you the details of the different types of spectrophotometers, but it's a handheld one manufactured by Thermo Fisher. It's less than a kilo, but it costs $65,000. So it's quite expensive. The advantages of spectrophotometers is that they are non-destructive. You can scan directly a tablet or even through a blister, even through plastic containers for some of them. So that's a big advantage that the Medicines Regulatory Authority like. And it doesn't require consumables like the mini lab, because the mini lab, one of the big issues the the regulators who are using Minilab have is to procure new reagents when they run out. And also in the Minilab, you need to procure reference standards that are quite costly and hard to procure. Okay. Another device that has been used in a few countries, but is still I would say under development. Maybe it's not the right term, but it's let's go through it and you'll understand. So it's a single-use device. It's called paper analytical device, or PAD in short. And it's very interesting. So it's a single-use device using colorimetry. So what you do is that you crush your tablet or you open your capsule of medicine and you spread the powder of it on a line here on the card. It has the side of a game card, right? It's quite small. And what happened is that you then put that in a bowl of water, normal water, and then by chromatography, some reagents that are within the card are going to migrate and meet the powder. And then there's going to be chemical reactions and there is a change of color in the different lines. And then what you see here is that you see a color pattern or a color code, and you then have to interpret this color code to see whether the active ingredients you are looking at is present or not. And that's just what the device, the card tells you. It tells you whether the active ingredients is there or not. It doesn't, for this specific card, it doesn't tell you any information on the quantity, but the developers have been working on another card similar to that can do semi-quantitation. And at the moment, they can test 60 different active ingredients, mainly anti-infectives, but they're also developing it for chemotherapy, for some specific chemotherapy. It doesn't cost too much money, but it's single-used and it's destructive. You need to crush your tablets. And some difficulties, and I'll come back to this, is to read the color code, but the developers have come up with an app recently that's still being tested to actually automatically read and interpret the results. There are many different other technologies, and I won't go through all of them, but I wanted to take you through a little bit of what we have been doing. And when I started to work on this topic, I first started to do a systematic review with the team on what are the different portable screening technologies available with scientific evidence that they work actually for medicines detection, medicines quality, substance falsified medicines detection. So we did that and published a review in 2018. And at that time, we found around 60 publications where researchers had tested the performance of 41 different devices. Mainly, half of them were spectrophotometers, but there was also the pad and other devices in that review. But what we found is that only a median of 2 active pharmaceutical ingredients, sorry, API stands for active pharmaceutical ingredients, only 2 active pharmaceutical ingredients were tested in these different studies, which is quite small when you think that there are like probably on the market now above 8,000 different APIs. And only 6 of these devices were, so we looked at portable ones, so only 6 of them were field tested. So there is a review published if you're interested more. And what we did at that time, we got funding from the Asian Development Bank and we were based in Laos still. And we did this multi-phase study with different collaborators where we used the review that I just mentioned to select 12 devices. We also discussed with experts in the field before deciding on the devices. And we tested 12 devices in a laboratory phase. Then, at the end of this laboratory phase, 6 devices were sent to us in Laos to do a near to real life evaluation with regulators using the devices, and I'll come back to that, of six of these devices. And in parallel, we conducted with health economists from the Moro network a cost-effectiveness analysis of these six. And at the end of this, we had a big stakeholder meeting with regulators from many different countries in Southeast Asia in particular, and other institutions. And we discussed the results, but I'll get back to this. There is 5 papers out of this work. If you are interested, I can send them to you. So what we did is that we looked at the 12 devices in the lab. It was a lab in the US, in Georgia Tech, the university. And what we did is that we tested the devices out as much as possible out of the box without doing a lot of settings and parametering, because we thought that was what would probably happen in a low and low middle income resource setting with potentially not a lot of expertise on these devices. And when we did that, we found that for falsified samples, so we made our own falsified medicines in that case. So we purchased a manufacturing press to make tablets. And we made falsified medicines containing no active ingredients, containing wrong active ingredients. And we tested the devices. We also tested them on some samples. We had real life samples, but these ones are sometimes very hard to get your hands on. So we had to make our own fake samples, but be assured that we didn't sell them afterwards. So what we found is that when you use these devices out of the box, they are really good at identifying falsified medicines from good quality ones, with one exception for one device that was a lower cost spectrophotometer. But besides that, they were really good. But for substandard samples analysis, so we also prepared substandard samples containing lower active pharmaceutical ingredients, lower amounts of active ingredients. And out of the box, they have a very limited success to identify them. And the most significant differences between the instruments were the setup and operation of the devices. So after that, selected 6 devices pictured here, and they shipped them to us in Laos, where we were based. We built what we call a simulated pharmacy in a room in the hospital where we were based. So it was made to look like a pharmacy in Laos, and we trained regulators, so the people who do the inspections of pharmacies in real life in Laos. We trained them to use these 6 devices, and we observed them while doing their inspection, their simulated inspection of our simulated pharmacy. And so what we found out is that there was a lower, kind of expectedly, there was a lower accuracy of the devices that need result interpretation, like the pads I mentioned earlier. There was difficulties with reading colors. We all are different to read colors. So, but this problem may not be the case anymore with the new app that the developers are currently testing, hopefully. But yes, they show a little bit less good accuracy. But for the spectrophotometers, there was not much difference. They were all good to identify falsified medicines. In that case, we had only falsified medicines in the simulated pharmacy. But there was problem with the reference libraries, and the differences between the devices. That was the time taken to test a sample. And there was also, we looked at the user satisfaction on each device, and there was satisfaction. I remember that regulators really liked devices that could fit in their bag, not the ones that with a big suitcase, because they travel by motorbike, or I mean, it's always more convenient in terms of logistics to have a small one. And they also particularly liked the spectrophotometer that I showed to you on the picture, that is is linked to a smartphone. So the result goes to the smartphone. They particularly liked this one. One interesting thing we found as well in that near to real life evaluation is that we asked the inspectors to conduct visual inspection as they normally do in an inspection of a pharmacy before using the device. But actually, when they had the device, they spent much less time to do the visual inspection of the medicines than when they didn't have it. So we did two different inspections. We asked them to do the inspection as they do in currently, that is without device, one with device, and we looked, but they were instructed to do the visual inspection, but they spend a lot less time, significantly less time to do the visual inspection. But that's a crucial step in identifying poor quality medicine. So that was a kind of a side effect of using devices, let's say. But that would have to be confirmed in larger scale studies, I would say. And so in parallel, we did a cost effectiveness analysis with health economists in Moru, and we found that so the cost effectiveness analysis was for if we were to implement the use of devices in the context of Laos, that's important to note. And we looked at these 6 devices, what would they cost to be implemented, and also the cost per sample. So to test one sample, the spectrometers are really cheap because don't take into account the cost of the device itself. But for the pads, it's a single-use device, so it's more cost, obviously. And the cost of implementation was from 5,000 to 2 million in the context of Laos, taking into account the whole country, this post-marketing surveillance of the whole country. And so what we found is that with all 6 devices we tested were cost effective if the prevalence of substandard and falsified medicine was high. But in cases where the prevalence was lower, then that was a different scenario. 4 of the 6 devices were cost effective. 2 of the spectrophotometers that the initial cost is high, like the TrueScan we saw on the picture, were not cost effective in that case. We have lots of caveats because there was lots of assumption behind these models, so we need to be careful. But that was one of the first cost effectiveness analyses done for devices. There are now, that study was almost 10 years ago, there are now a few more showing good results, especially for the pads. Yeah, and in both scenarios, the near SG1, which is the one with the smartphone that I mentioned already, was the most cost-effective option. It's a low-cost spectrophotometer that costs around $2,500. That's the one, sorry. But yeah, again, we need to be aware of the limitation of this study. And then after that, these 4 different steps, phases of the study, we conducted a big stakeholder meeting in Vientiane, and we presented the results to regulators from different countries, and we had hands-on sessions with the devices, and we discussed what they think about, because we had the example of Laos, but not the rest, not other regulators. And we discussed different aspects around the legislation, around devices, logistics, what would be the problem to implement them, etc. So that was a very interesting 2 days meeting. And we published 5 papers on this study, and one includes recommendations on what to think of when you want to implement the use of a portable screening technology. What do you need to consider? And we had different aspects. I won't go into all the details, but yeah, there's lots of details in the article. Another work we are doing in the Medicine Quality Research Group and with a consortium that is called the V Consortium, that stands for Vaccine Identity Evaluation. that started during the pandemic, when the pandemic started and we knew that vaccines were being prepared. We knew that there would be issues with potential falsification, substandard vaccines, but also degradation because of the cold supply chain that needed to be maintained. So the head of our group, Paul Newton, gathered together a team of chemists, biochemists, physicists from the university and from the Rutherford Appleton Laboratory in the south of Oxford. And we started to look at repurposing existing devices to test COVID vaccines and other vaccines. In the end, we tested a few different vaccines. And one technique that was suggested is the MALDI-TOF. The MALDI-TOF is a laboratory device. We're not talking about portable right now, but that is used in many countries, in many microbiology laboratories to identify bacteria and bacteria species. And the idea was to use it for vaccines. We also used the SOURCE, which is a spatially offset Raman spectroscopy. And this one is one that is used actually in airports, airport security, where security staff check whether there is explosives in liquids that you transport, that you carry, for example, for babies. And there is a portable version of it in the airports. It's a bit less portable. It's not so big, but less portable. But there is a portable version of it that's on the picture here. So we tested this one as well for vaccines. What is good is that it's as many spectrophotometers. It can scan through a vial, a glass vial. So that was one of the big advantage of this one, whereas the MALDI-TOF is destructive. You need to open the vial to be able to test the vaccine. There was also brilliant ideas to test lateral flow assays that everybody has had many during the pandemic, I'm assuming, to actually test the quality of the vaccine directly on the strip. And we also tested other brilliant ideas, low-cost techniques that are used in biochemistry or in different labs, like urine strips, ethanol strips, cholesterol meters. So all this could be used. All these techniques give you different information on a vaccine. It doesn't tell you the whole story of the whole. It doesn't check the whole potential defects that can be found in a vaccine, but in combination, these devices could be very interesting, and they are either affordable or already available in many places around the world. So that was the idea of this. A few papers have been published. I won't go into the details right now because I have only a few minutes left, but if you're interested, you can visit the webpage at the bottom and or ask me if you would like the papers. And some of you may have heard of the recent incidents of intoxication with diethylene glycol and ethylene glycol that was present in syrups that resulted in the death of hundreds of kids in different countries, Gambia, Cameroon, Indonesia, Marshall Islands, Uzbekistan. So diethylene glycol and ethylene glycol in syrups is not a new thing. It's been happening since almost 100 years. The first case actually happened in 1937 or 38 in the US, and there was intoxication. There was contamination with ethylene glycol and the ethylene glycol in antibiotic syrup, and I think 100 persons died at that time, and that actually led to the reinforcement and strengthening of the US regulations on medicines. And there has been many cases since then. But very recently, there has been more than 200 that we know of death of children below 5. because they were taking paracetamol and cough syrups contaminated with that very toxic renal toxin that is diethylene glycol and ethylene glycol. In these different cases, there was a mix between factory errors but also fraud, because the diethylene glycol and ethylene glycol are products that are used in other industry. They are used as antifreeze and brake fluids for cars, and they are cheap, but they look like and they smell like and they taste like an excipient that is commonly used in syrups that is called propylene glycol. And you really cannot, it's not easy to tell the difference between the two, and that's been used in the past as a cheaper option as solvent in syrups. So when that started to happen, the recent case was in 2022, and many kids started to die. We thought that we could use the devices that were used for the V project that I presented in the previous slide to actually detect diethylene glycol and ethylene glycol in syrups, because the reference method in the lab is gas chromatography. Gas chromatography is not available everywhere, not at all. There is not a lot of labs that can do that. It's costly, it takes time, and you need a lot of expertise to run it. And so we thought, okay, should we have screening technologies to look into this issue? And so we use the similar technologies that I presented in the previous slides. We had good evidence with the MALDI-TOF, and we are still working on different devices, but papers will come out soon around this. Okay, I'm gonna stop talking about all the studies we've been doing. Just thought I would mention that. So from the work we've been doing, there is quite a lot of evidence gaps, especially technical on related to technical performance. So what device can do what or cannot do what, and they are listed here. One concern that we particularly have is that people who are regulators who are already using some devices may think that these devices may be telling you everything, but they are not actually. As we showed in the multi-phase study, they are really good at detecting falsified medicines if they are correctly parametered, but less good with substandard medicines. Yeah, that's a concern we have, is that there are misinterpretation or misuse of them. And then, as I mentioned earlier in the systematic review, we found only few active ingredients were tested when there are 7,000 globally. There is not a lot of work for devices, especially spectrophotometers, to detect falsified tablets that are coated, because the coating can interact with the signal, and there is a lot of different gaps. I'm not gonna talk about them all, but one thing that I wanted to come back to is the problems with infrastructure support and implementation of these devices. So I mentioned the reference libraries, and I explained to you how they are supposed to be built. So you may remember that reference library that are to be used for spectrophotometers needs to be made with genuine medicines coming from the manufacturer. And that's quite tricky to get samples from manufacturers, even for regulators. It's tricky, it takes time, there are issues with pharma companies. And in a study we did recently, actually, where we looked at the quality of antibiotics in different countries, we really struggled to get from some manufacturers. I think we got probably, if I'm not mistaken, like only 40% of the one of the different brands we collected, we only got 40% of the genuine reference. So that's quite complicated. And in real life, for post-market surveillance, who should be responsible for creating this reference library? So that was something we discussed in the big stakeholder meeting. There was no common agreement on this. Should that be a regulatory agency, so each national regulatory agency doing that, or more a regional approach, and how to set this up in practice? It was suggested that roll up to use the devices only on a few brands initially, because that's more doable, more feasible. But there is no big solution on this. The WHO is working on that, but it takes some time. And also, how will library be stored? There is problem around data security, and also it needs to be updated. Anytime a manufacturer change anything in the formulation, then you need to get a new reference library for it. And also, we had this case in our studies that we got the medicines from the manufacturer, but we tested it with laboratory techniques to check whether it was good quality actually, or of expected quality, but it was not, because it was substandard. So that costs money. It costs money to test the quality of all of medicines. So that's a lot of implementation issues around this. Also, there is not a lot of evidence on the training, what training is required for end users of these devices, and even for less end users, the ones who set up the devices. Cost effectiveness analysis, not a lot of data. False reassurance of devices. That's what I mentioned about. That's what I mentioned just before. People may think that these devices, because they look fancy, they are expensive, they can tell you everything, but they actually cannot. And also, which device to use at what level of the supply chains in different community and contexts. There is not a lot of work on this. And yeah, so there is, so regulators are willing to use them, these devices. But there has been a report recently from the WHO last year, where they asked regulators what are the challenges or obstacles to actually implement these technologies. And only 27 responded, but I thought it was interesting to see that, yeah, they have not enough technical knowledge to know how to develop the system. But also they are confused because of all the variety of devices available on the market, what device should be used and at the lowest cost. So in our group, we decided to develop a tool. It's an online platform to help regulators to make decisions on the best devices for post-market surveillance. And so I'm just gonna, that's almost the end of my talk. Yeah, I'm gonna try to speed up a little bit. But it's a resource that we have developed online. It will be made available soon. We extract data from the literature, from that review that I mentioned, and we've updated it now. And we have developed what we call the dashboard, Daffodil. That stands for Dashboard for Field Portable Screening Devices Using Scientific Literature Evidence. How we developed it, we gathered regulators, so potential end users of the dashboard, together in different events. And we have developed a prototype of it. And then my colleague Christian Hagel is now conducting a user experience study. And Daffodil is meant to be live in July this year. So just a quick tour, what is Daffodil? So it's a dashboard. When you come on the welcome page, the landing page, you will see a few charts, a few information on what's in the database. So in that case, it was 58 devices. It's not up to date, it's much more now. Also much more scientific publications, how many active pharmaceutical ingredients were tested in these different studies, these types of information, sorry. And then if you go to the device page, you can filter out, sorry, that was a mistake. Yeah, so you can filter out based on different filters. So say you want to have a device that is very small, that is already marketed, because in the dashboard we also show devices under development, and that is battery powered. So you click on the different filters and it will show up in this table. You have a default number of information and you can add additional ones by clicking on a list of variables of data available. Say we want to add the formulation, it appears on the table, easy one. And one nice feature is that you can compare between devices. So you will select up to 5 devices by ticking these boxes and then click on the compare to selected devices, and it will show to you all the different characteristics in parallel for the different devices. What else? And you can also access the original publications from the dashboard. Okay, just want to finish 2 slides or 3 maybe on other technologies because we talked about detection technologies, especially portable ones, a lot. But there are other technologies that are used in the medicines quality world. And we are conducting a project on testing these technologies to identify the origins of falsified medicines. So these ones are more laboratory-based techniques. But how the idea came from is that in the late 90s, I don't know if you remember one of my first slides where I showed to you genuine and falsified packages. There was this medicine, the artesunate tablets. And I told you there was a big epidemic where more than 80% of the pharmacy would have falsified artesunate tablets containing no active ingredients in Southeast Asia. So the head of our unit at that time, Paul Newton, at that time based in Laos, started in collaboration with the WHO, Interpol, the pharmaceutical company who is actually making these medicines. I mean, the genuine medicine of it, and other partners. They collaborated to use different techniques. So they did packaging analysis, they did Pauline analysis, and they did stable isotope analysis of the medicines they found in the different pharmacies. And interestingly, they found so they looked at all these different data, also chemical analysis of them, and they looked at all the different data, and the evidence seemed to say that there was 2 different types of medicines in there. So these are the 2 different sets coming from, found in different countries. But when looking into more details in the results, they also found pollen, especially of Pinus and juglans, found in the tablets. And altogether, the analysis and the results led to good clues where some people were trading on these medicines. And it led to the arrest of a few people in collaboration with Beijing. So that was a very interesting one. The pollen analysis was a key part in this investigation. Unfortunately, there is not a lot of forensic palynologists in the world, actually. Many of them are already, there was a few of them at that time, and they are all retired. We didn't identify new ones. But the idea was okay, well, we cannot use pollen, but what about using environmental DNA analysis? So the idea came from there, and that's when we got Paul Newton got a grant from the Wellcome Trust, and to do this project we call FORESFA, Forensic Epidemiology and Impact of Substandard and Falsified Antimicrobials on Public Health. That is coming to an end, actually, but I'm not going to show you results today because the results are being discussed, not yet finalized. But it's basically a very innovative and multidisciplinary research project with specialists in pharmacy, public health, forensic genomics, chemistry, archaeology, social network analysis, and modeling work. There are different aspects in this project, 2 main streams, let's say, but I will only show a part of the one main stream of interest for this talk. And the aim of this was to gain information on the origin and trade routes of falsified antimicrobials. using forensic techniques. And these techniques, eventually, if they work, could be used by regulators, enforcement, academia, and pharma industry. So this is a big consortium of the FORESTFA team, quite a global team. Also, if you want more information, there is a web page on the CTHR website. And so what we're doing is that we are looking at the environmental DNA. We had also a pilot, a proof of principle from the past that was done by the team. And we are looking at the eDNA can provide information on the environment and origin of the tablets, capsules, and excipients, and potentially on the time of manufacture. But we have already found bacteria, fungi, plant, and even human DNA. And we are currently analyzing all this data, but we're also looking at the more chemical approach using stable isotope ratio mass spectrometry. One interesting thing is that in the tablets, in the medicines, there is water. Water is used for the manufacture of medicines. And I didn't know before working on this, but there is the stable isotope ratio the stable isotopes ratio differs in the water based on where the rainwater comes from. And you can see this from this isoscape on the screen. And so this can give you information on where where the water that was used to make the fake tablets came from. So this is the idea, but we're also looking at other isotopes from different excipients used, like different types of flour and other excipients used in tablets. So exciting work. There will be some publications soon, but welcome to send me any questions if you have or to discuss now. Thank you very much.
Hey, thank you.